A superneutral is a type of antibody that exhibits a unique binding characteristic. Instead of targeting a specific antigen like most antibodies do, a superneutral binds specifically to the Fc region of all antibodies belonging to a particular species. This means it can interact with any antibody produced by that species, irrespective of the antibody's specific target. Now let's delve into the applications of superneutrals within the field of immunology. Firstly, purification of antibodies. Superneutrals offer a method for purifying antibodies from complex mixtures. Because of their ability to bind to the Fc region, a superneutral will selectively bind to antibodies present in a sample, leaving other proteins and molecules unbound. This selective binding enables the isolation and purification of antibodies. Secondly, detection of antibodies. Superneutrals are useful for detecting the presence of antibodies in a sample. By labeling the superneutral with a fluorescent or radioactive tag, it can be used as a probe to identify and quantify antibodies. When the labeled superneutral binds to an antibody in the sample, the tag becomes associated with the antibody, allowing for its detection through fluorescence or radioactivity measurements. Thirdly, blocking antibody binding. Superneutrals can be used to block antibodies from binding to their specific target antigens. This is achieved by pre-treating the antigen with a superneutral. The superneutral binds to the antibody and prevents it from interacting with the antigen. This can be useful in situations where it is desirable to inhibit antibody-antigen interactions. Finally, in vivo antibody depletion. Superneutrals can be used to deplete antibodies within a living organism, or in vivo. By injecting a superneutral into an animal, it will bind to circulating antibodies, neutralizing them and facilitating their removal from the bloodstream. This application can be beneficial in situations such as autoimmune diseases or other conditions where reducing antibody levels is desired. In summary, superneutrals are a valuable tool for studying and manipulating the antibody response. Their versatility and broad applicability make them useful in various immunological applications, from antibody purification to in vivo antibody depletion.